I'm going to tell you about what I'm calling neural circuit theory. But before we actually even get into that, these are some of my collaborators on this. Wayne Mackey was a postdoc in my lab and then founded this video game, neuroscience-based video game company. Any gamers in the room? Anybody? Have you ever heard of AIM Labs? AIM Trainer for first-person shooter games based on neuroscience? Well, check it out. That's that. This guy is a very close friend and colleague in the physics department at NYU. And these are some of the guys that we're advising together, all super smart, super brilliant people. I tried really hard to not follow in my father's footsteps and be a physicist, but you can't help it. So here I am. So the brain is a complex dynamical system. What do I mean by that? It's not just complicated. I mean that in a formal mathematical way. This is a diagram of just some of the brain areas at the back of the brain involved in vision. V1, primary visual cortex, creatively V2, secondary visual area, V4, so on and so forth. They all have names. They're all identifiable. I'm not going to talk about that much today, except to point out that the size of the rectangles indicates the relative surface area is proportional to the number of neurons in each of these brain areas. And the size of the lines is proportional to the strength of the connection, the number of axons connecting the two. So there's a strong connection from V1 to V2, V2 to V4, from V1 to V4. But for every one of those, there's also a feedback connection. So there's a reverse connection from V2 back to V1, and from V4 back to V2, back to V1, and from V4 back to V1. So there are these recurrent long radius. And then if you zoom in and look at the circuitry within any one of these brain areas, there are lots of different cell types that are interconnected in particular circuit motifs, and recurrence is the theme. Okay, there's strong recurrent interactions between them. Lots of different types of neurons, lots of synapses. So it's complicated, but it's also, in the mathematical sense, a complex dynamic system with recurrence and feedback. Right? So what that means is that, it's hard to predict what the outcomes are going to be. You can try to manipulate or perturb the activity in a neuron or a subpopulation of neurons, but that's going to have a ripple effect throughout the brain that's going to cause feedback onto other neurons that are right nearby where you think you have control over stuff. And it's actually really, really hard to predict what the outcomes are going to be. And so that's why we need a theory. So neuroscience, and I'll argue AI, need a theory, kind of like Maxwell's equations for the brain, right? Expressible in mathematics, hopefully analytically tractable, or at least partially analytically tractable. Is that Matt Turk? Matt. Oh, God, it's been forever. Okay. All right. So why does this matter? Well, for neuroscience, it matters for reasons that I was just telling you, right? Right now, every brain-computer interface is an experimental system. Every attempt at developing a drug for a degenerative disease or a developmental disorder is guesswork. If NIH were to fund the development of a new airplane wing, they would design it based on a hunch, put 300 people on the new plane, 300 people on a control plane, and count how many people died. This is not how a mature science works, right? If you're actually an aeronautical engineer, you design the airplane wing, you do some computations on a computer with software that's widely available, and that's it. You're done. It works. You know that it's going to work, right? And so I think we're at a point in time at which we can actually do this in neuroscience, right? Or get close to it, or try to work toward it. Now's the time. And the same is true for AI, right? The human brain operates on 10 watts. Sam Altman is building gigawatt power plants to power his AI systems. And we all know that, the development of AI over the last decade, there's some cool insights, but a lot of it has been thousands of people coding and tweaking and, just trial and error. All right. So let's move beyond trial and error. Why might that be possible? Well, because in fact, all these different types of neurons are connected into, sort of canonical microcircuits. You look at any little chunk of brain, there are differences, but the differences are subtle and the similarities are strong. So there are particular types of neurons connected in particular ways that perform what we think are a particular suite of canonical computations. If we understand that circuit function, so we in a way that we can write it down in a system of equations and handle it in an analytically tractable way at least some of the time, then we should be able to solve lots of problems up here and also some problems here. That's the agenda. Okay, so this is where we're, this is the proposal for today. This is as far as we've got. And I called it organics. For those of you who know who my Nobel laureate dad is, he was awarded the Nobel Prize for organic conductors. So now when you Google Heeger organics, you get the so that's the real reason for all of this. Okay, that's actually what it's all about. But it stands for oscillatory recurrent gated neural integrator circuits, and you'll see they oscillate, they're recurrent, they're gated, they're neural integrator circuits. So I'm not lying. Okay. Right. So, and here's the basic idea. So the principal cells, which you can think of as like layer 5 pyramidal cells, for the you who know that about neuroscience, follow the membrane equation. Tau dy equals minus y is like the membrane potential plus stuff. The stuff is the synaptic input. And I'm just going to refer to it as input gain times input drive plus recurrent gain times recurrent drive. Later on in the talk, I can show you how this could be laid out in terms of the dendritic tree and biophysically plausible mechanisms for the time being. All right. So what is the input gain? Well, that's a modulator that gets multiplied by the input drives. It's going to come from another subtype of neuron. What's the input drive? Well, the input drive is a weighted sum of the inputs. So you have a collection of inputs. Let's say we're in V1, primary visual cortex. This is our principal cell. These are the inputs from the lateral geniculate nucleus of the thalamus. I compute a weighted sum of these inputs, standard receptive field model that you teach, intro perception or intro neuroscience, right? Linear receptive field. It gets multiplied by this gain, which later is going to play a role in attention. And that's one of the inputs to this principal. That's this stuff. The other one is that the outputs of these neurons have a recurrent drive onto themselves. Okay? Weighted sum of the outputs. And that weighted sum of the outputs is multiplied by a different modulator, different gain control, okay, coming from a different cell type. And that is determined by this outer recurrent loop. Okay? So the outputs of the principal cells go through the other cell types, and then these guys kick in to control the gain of this local, of this in here. All right. So two recurrent loops, one around the other. And the idea is that this one recurrently amplifies whatever came in through the input. Okay, strong recurrent amplification. And this one shuts that down. So if there's a weak input drive, you get lots of recurrent amplification. If there's a strong input drive, then this starts to recurrently amplify, but it gets shut down by this outer. Right? And if this outer loop does the right form of computation, I'm not gonna, I'll just hand wave here for a moment. All right, then this whole system has this thing as its steady state. Those of you, and I'll go through this in some detail in a minute. This is called the normalization equation.
I had.
Right? And what it looks like is the output firing rate is proportional to the input drive, half of vector squared, divided by a constant plus a weighted sum of other input drives. And I called it normalization because basically what you're doing is you imagine a vector Z of input drives, and you normalize the length of the vector. Okay. Right? This thing squared divided by sum of the things squared. If all of these weights were 1 and that were 0, it would literally be norm squared of the vector. You have the back.
Just to get an intuitive picture, what would sigma and W depend on in the XYZ be Y and A?
Yeah, so we'll see some of that, right? So sigma is going to determine the contrast gain in visual cortex. Okay? It's going to determine the rollover of the saturating nonlinearity. Okay, and this thing determines essentially the normalization pool, right? So what's being characterized here is each neuron's underlying sort of receptive field, right, is being suppressed by a weighted sum of the other ones. Okay? And that weighted sum is the normalization pool. If you want to think about this from sort of a neuroscientific point of view, you know, each neuron at each location in cortex is suppressed by a whole bunch of other neurons that are right nearby. That includes, like in visual cortex, neurons that are selected for different orientations, different spatial frequencies, right?
So can I ask?
Yeah.
So here the dynamics are only figuring out in terms of applying some sort of nonlinearity and normalization and such like. They're not really playing any role in terms of temporal information transfer or anything like that, right?
But they are,
Yeah.
because this is also, you can think of this thing, and we'll get to this, right? So this recurrent drive here can do lots of stuff, right? Here I just described it as recurrent amplification, but we have separate control over these gains. If I were to set this gain to 0 and that gain to 1, then this behaves just like a feed-forward, say, convolutional neural net. If I set this gain to 0 and this gain to 1, then I have a working memory circuit. Okay, it'll hold the concurrence a representation of, you know, whatever the input was previously for as long as you want, right? And one of the key ideas here is that the normalization stabilizes that recurrence, which is a big problem both in brains and in AI. And instability in brains, you know, would mean that the organism dies. Right? Little bits of instability means things like epilepsy and autism, right? And in AI systems, you have the same problem. If you have an unstable circuit, right, let's say it's oscillating, then I dare you to try and get machine learning to work, right? It would be like trying to fit a line to data in which the data points are moving around, okay? You have no stable signal upon which to learn. Right. And this is a big problem in recurrent neural nets, and part of the reason, I think the main reason why people have sort of abandoned LSTMs and GRUs and gone to transformers. And as soon as you go to transformers and build these language models with 1,000 input tokens and trillions of weights, then you need to boil the ocean and build gigawatt power plants. Okay. But if you had a stable recurrent circuit, right, that didn't suffer those problems, then we should be able to get the same level of performance out of recurrent neural net that people currently get out of transformers with far fewer parameters, no input buffers, you know, 10 watts instead of gigawatt. That's the goal. Okay. All right. I gave the whole talk. All right. So, and in fact, here's the summary. So I wanted to put this up front in case I don't get through it all. So we're going to take this basic circuit, we're going to stack it from one brain area to another. We're going to add feedback to it. It's going to be hierarchical with recurrent feed-forward and feedback connections, such that you get this normalization operation to happen within each brain area, within each circuit. And it's analytically tractable. We can linearize around the fixed point because it has a known fixed point that was the little equation on the previous slide, right? That's our attractor. We linearize around the fixed point and derive closed form expressions for things like the power spectrum, the cross spectrum, from which we can derive predictions in closed form, not through simulation, but through evaluating function. For things that neuroscientists measure and psychologists measure, like the modulation from attention in both behavior and neural activity, oscillatory dynamics in the power spectrum, and the coherence spectrum between brain areas, what neuroscientists call communication subspaces, and dynamic control of functional connectivity. And noise correlations. That's all because we have this analytical tractability on this thing. We can do a stability analysis, again, because it's analytically tractable. We can understand the parameter regimes in which it's stable, in which it starts becoming unstable and then goes unstable, right? And avoid those. And we think that's also useful for understanding things like epilepsy and autism, and other neurological diseases. in which there's evidence for failures of normalization and failures of stability. I'm going to show you that on some simple benchmark, we can get state of the art or near state of the art performance. And if there's time, I'll show you how we can implement it physically. Okay? That's the whole talk in one sleep. All right. Quick review. Those of you who haven't seen it before of what normalization is. This is a view of the bottom of the brain. Eyes are in the front, primary visual cortex in the back, without trabecular nucleus, thalamus is in the middle. You take a look at the neurons in primary visual cortex, and we'll go look at other brain areas in a minute too. And you know what Hubel and Wiesel discovered and what they were awarded the Nobel Prize for in part was, you know, the discovery of orientation selectivity, right? That everybody knows this. Selective receptive field is just weighted sums of the inputs. Positive weights here, negative weights there. You put a vertically oriented stimulus within that receptive field, you get a big output. You put a horizontally oriented stimulus within that receptive field. The positive weights cancel with the negative weights. You get no output. Likewise, if there's no feature in there, you get no. So that's our input term. Weighted sums. All right. And then you have to imagine that this is being replicated across space, across channels, or I guess in machine learning, we call them feature maps, with different orientation preferences and different spatial frequency selectivity. So if this is your input image, you know, you get like high spatial frequency verticals, high spatial frequency horizontals, and likewise low spatial frequency vertical diagonals, and so on. And that was sort of the story in the late 1960s and early 1970s. About how primary visual cortex encoded information about the visual. Okay. Here's another way of looking at that. And I'm going to be making a particular point here in just a minute. Instead of, this is no longer the tuning curve for one neuron. This is now what I call a population response. So when you measure the tuning curve a couple slides ago, you record the firing rates of an individual neuron as you vary the stimulus, say, orientation. Now, if you hear what I'm doing is I'm imagining a thought experiment that I record simultaneously from 3 different neurons, all to the same input stimulus. One of them is selective for vertical orientation, so it gives the biggest response. This one is selective for slightly tilted this orientation clockwise, and this one tilted more counterclockwise, right? So we can, you know, you could imagine now you can record like 15,000. It's fine. So you have a bunch of ones on this, right? The point being that the neurons together represent information that you could decode about the stimulus. You could read out what the stimulus orientation was, either as the peak or as like the mean of this distribution. And if I tilt the stimulus a little bit, I change the representation a little bit. This one goes down, that one goes up, this one goes down. I could still read off where the peak is kind of the mean. You can think of this as, you know, normalized, like a probability density, right? Then, you know, sum of height times preferred orientation gives you a readout. Right. And of course, these neurons code multiple things at once. So if I change from a high contrast to a low contrast image, I just scale this thing down, right? This is all linear up until now, just weighted sums. Scale the input, scale the output. I cut the contrast in half. I cut all the responses in half, but I can still read out the orientation as well. Okay. Now, why did I go through all of it? Well, so in brains and in machine learning, the next thing that was added is a rectification. And this was done in the neuroscience literature in 1978. Moffitt, Thompson, and Tolher, 78 actually invented the rectified linear unit. Okay. So if you do that, more reasons. Simple idea, right? Firing rates can't be negative, whereas the output of this thing can be. So I draw a rectification on this, okay? And then I can encode the positive, what were the positive and negative values by having a complementary pair of receptors. It's easy enough. Wherever this one had positive weights, the other one has negative weights, so forth. Okay. And if you want to, you could put a saturating nonlinearity on that as well, which is constant. All right. But it turns out that that's just wrong. There's something that's, and now we're in the kind of 1970s, late 70s, early 80s, okay? Early to mid 80s. And what people observe is that as you, first in cat and then later in the cat monkey, as you increase the contrast of an input, if that were all true, then the responses should just scale up with it. So you double the contrast from here to here, the responses go up by almost a factor of 2. But when you double the contrast from here to here, the responses barely go up at all. And that gives you this roll off here, this response saturation, right? Which in machine learning would be like throwing a sigmoid or a hyperbolic tangent on the output in addition to. And that's a problem. It's a big problem. This is what we had before. Now imagine I have this saturated. Okay. Now I try to turn up the contrast and I hit the saturation, right? What that means is that the output activations in my machine learning system or the output firing rates of my neurons are going to hit a ceiling. And instead of having this nice, bell-shaped curve from which I could read out information about the stimulus, I now am capped. And I can no longer discriminate orientations at high contrast. That's wrong. We know psychophysically that if anything, you get better at discrimination, higher contrast. So that's wrong in that sense. But it's also just fundamentally the way that neurons and artificial, neurons and an artificial neural net encode information is in terms of the relative activation, right? This one gives the biggest response, then that one doesn't. Right? And having a sigmoid or saturating nonlinearity on the output destroys that representation, right? It's the relative responses or the ratio of the responses that matters, right? And by throwing a sigmoid on the output of your artificial neural net to try to keep it stable, keep the activations from exploding, you're destroying the relative responses and messing up the representation. It's the wrong way to do it. Okay. So that's a problem. That's not how it works. In addition to that, in the brain, there's another dramatic failure of that story, which is a failure of additivity, right? For a system to be a linear system, even with a rectification in there, it has to scale and it has to add. This is an example of what happens if you take a stimulus that gives you a big response and a stimulus that gives you no response, you superimpose them and you end up not with the sum, which would be this plus 0 should be this again, but about half that big. Okay. So there's a mutual suppression going on somehow. What originally this was called cross-orientation inhibition. And again, here's a bunch of curves showing you the firing rates, a function of contrast, when the preferred orientation, when there's no orthogonal mass presented, and as you increase the contrast with that orthogonal mask, and what this looks like with a shift to the right on the log contrast axis, which is the signature of the kind of thing. All right. So that's where this normalization idea comes in. Right? Initially, I introduced it sort of in this form in the late 80s, early 90s, right? But the overt firing rate isn't just a rectified or sigmoided version of the underlying linear weighting function. Instead, it has this built-in gain control in which the input drive to that neuron gets divided by a sum of all of the neurons. Right? And this has been adopted in artificial neural nets over the past few years in exactly this form, in fact, and called normalization. There was the first layer normalization. This is called filter normalization. Okay? In the AI literature, but it actually dates back to. Right. And what this does is for a preferred orientation stimulus, again, we're in visual cortex, the responses increase, saturate. Why do they saturate? Well, you increase this value, one of the value, this guy in the numerator, you're also increasing the denominator as well, right? So eventually Z gets much bigger than sigma, and this is going to just be Z over Z, or Z squared over Z squared. For a non-preferred orientation, it's just going to scale down. Okay? Maintaining the ratio of the responses throughout the whole. And when I superimpose a second stimulus component or an additional feature, right? That doesn't drive the numerator, it still drives the denominator. So that's going to shift the cursor. And now you can see, for whoever asked that question earlier, sigma is literally the semi-saturation contrast. It's the contrast that gives you half the maximal response. Okay? So it's in units of contrast in visual in primary vision. Elsewhere, you're going to lose track of what those units are, but at least in a sensory area like that, you can specify. Now, this turns out to be an important idea, more important than I thought it was at the time. Right? This does a bang-up job of explaining these nonlinearities and the responses of neurons in V1, but we've learned over the last, good God, 40 years, that you see this same kind of computation happening in every brain area and every sensory, perceptual, and cognitive function that anybody's ever tried to look for. Okay? You see this in a bunch of different visual cortical areas. You see this in working memory. You see this in we're about to publish a paper on navigation and head direction cells. You even see this in radically different neural systems in different species. This is an example from fruit fly olfaction from Rachel Wilson's lab at Harvard from a number of years ago, showing you the same shape curves, the same shift. Now you're superimposing two different odorants at the same time. You get these curves to shift over, fit with the same equation, except that the exponent, for reasons I don't know, in the fruit fly olfactory system, instead of being 2, which is nice, where it is nice, it happens to the best fit exponent is 1.5. Fruit flies don't know about it. Okay, so as I said, also lots of different visual areas, other sensory modalities. Oh, this is a cool one. My friend and colleague at NYU, Paul Glinter, has shown that you get exactly the same form of computation in subjective value representation in parietal cortex and medial prefrontal, which explain these paradoxes of behavioral economics, a number of phenomena that have been observed by behavioral economists, and other examples. And I'll show you, we've also applied students about that. Okay. Working memory control retention. So why bother with this? Well, that I already said. Limited dynamic range. You don't just want to saturate because that throws away the information. Instead, you want to gain control. And that's what this does. Bunch of other ideas. Here's the most recent idea. In terms of the dynamics, this stabilizes the circuit. And it's kind of intuitive that it should. That it's going to prevent explosion, it's going to prevent the circuit from exploding by having this built-in gate. Every single electronic device you've ever had built-in gate. Right? So get started. All right. There have been over the years in between 1990 and recently different ideas for how normalization might be implemented in the brain. There are various reasons why all of these are wrong. The key idea that's been observed now repeatedly is that instead of, so remember, this is the steady state. This is the attractor state that it ends up in, right? The dynamics of this thing will end up here if you give it a constant input. Okay? But what's written here, the brain can't do this literally. And artificial recurrent neural net can't compute this up front because these things are hidden variables. This is the input drive. Right? This isn't represented in the brain as an overt firing rate. Okay? And if you were to try to do this calculation in kind of a straightforward way in an artificial neural net, well, the Z's could be unstable. Right? So you can't, it doesn't work in a recurrent neural net to say, all right, I'm going to have my potentially unstable recurrent neural net here and then a layer that does normalization. And then my next layer of recurrent neural net, the way that you would in a company, right? Because the recurrent neural net can be unstable. So by the time you get to normalizing, it's already too late. The normalization has to be built into the recurrence. Yeah.
Is Z the stimulus X?
So the input to the circuit is X. In visual cortex, this would be like contrast. The Z is a weighted sum of the X's. the input drive. So that's my receptive field, or in an artificial neural net, that's my embedding matrix.
So these are the.
Yeah.
And what do you mean by that, four symbol operators?
Yeah, half wave rectification and square. Okay. All right. Okay. So what we've learned about how normalization works in the brain is that instead of thinking of it as being literally division like this, the way that it works is, as I was describing earlier, stopping the recurrent input. So the intuition is multiply by less is the same as divide by more, right? So this does a recurrent amplification, multiply, 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 stop it. Okay. So that's kind of the core. All right, so great. So now we can run some simulations, different parameter values. Again, I'm in modeling visual cortex here. At low contrast, you get these slow ramp ups, and then it reaches the steady state given by the circles. At high contrast, you get these overshoots, and then it bounces back. You see things like this in electrophysiological measurements in the primary visual cortex. Under some circumstances, you can get what looks like oscillatory activity in the gamma frequency range, something that I'm going to come back to over and over again over the next few minutes. We don't think that there's actually literal stable oscillations like this, like a Hopf bifurcation for those of you analytical systems, but rather that it's sitting in the spiral attractor regime. For those of you who don't know what I just said, you will know it in 5 minutes. Okay. All right. This is one of the key measurements that supports this idea of recurrent amplification. So this is a simulation of the membrane potential fluctuations, okay, of that circuit. And this is a simulation of what that looks like if I shut down the recurrence. Okay, so I literally just turn this off. Okay. And the thing I want you to notice is that the range of values here is a factor of 10 smaller. Range of values there. David Furster at Northwestern did this experiment, good God, like in the 1990s, and in the real world. And the way that he shut down the recurrence was with a cooling probe on the surface of the brain, which shuts down the action potential. So the input was still there, but there was no recurrence because there was no action of the neurons in the local circuit in the cortex. And he observed essentially exactly this factor of 10 decrease, but retaining, sensitivity to increasing contrast, retaining orientation selectivity, spatial frequency selectivity, all of that, just shutting down the recurrent. All right, we can derive stuff in closed form. I'll skip that. I want to show you this to just introduce these dynamic systems ideas for those of you who don't already know it. So there's 3 different types of neurons in this circuit. There's my principal cells Y, and then I'm calling them A and U, the outer loop that does the recurrent. Okay. If I have a low contrast stimulus with a relatively weak input drive, and I initiate a simulation starting all 3 neurons at 0, it jumps right to the fixed point. Okay, so this is three-dimensional. Okay, it jumps right to the. For a kind of medium strength input drive, you get into this what's called spiral attractor regime, in which the responses overshoot and then spiral in the fixed level. So if I were to be looking at just the principal cells, the Y neurons, it would look like a damped oscillator. Okay. All right, and that's what I mean by the spiral regime. Under some circumstances, with certain choices of parameters, you end up with stable oscillations. And that's what's shown here. If I start from 0, it starts to spiral in, but then it just gets stuck here in what's called the limit cycle. Likewise, if I were to start it from the fixed point and perturb it a tiny bit with a little bit of noise, it actually spirals out to the cycle and stays there. So you get stable oscillations. Think about what's happening in the Y neuron as it's going around this limit cycle. Well, Y goes up, then it goes down, then it goes up, then it goes down, then it goes up. And that's where you see these stable oscillations. This is actually like a potato chip shaped bend. That's interesting. It's not a linear system. All right. Now, what we think now is that neural circuits, stay in these two regimes most of the time. And what people report seeing experimentally as gamma oscillations is actually this with noise added. So imagine what's going to happen here. If there's no noise, it spirals into the attractor. But if you add a little bit of noise, occasionally it's going to push it out. The noise is going to bump it out and it will spiral back in. So you get these bursty, occasional periods of oscillatory like activity, which is a nice description of what gamma oscillations actually look like in the experiment. Okay. All right. So and I'll come back to that. So as promised, we can do an analytical analysis. We can do an analysis. I can't, but my colleague Stefano and his brilliant advisees can, of the stability. For those of you who are like deep in with me here, the color map here is the largest eigenvalue of the Jacobian of the linearized dynamical system. What I'm doing here is increasing the input drive. That is contrast in visual cortex, or whatever strength of input to some other brain area. And on the y-axis is a measure of the recurrent interaction strength. Okay. Now, earlier when I told you that it achieves normalization exactly as the fixed point, I was sort of lying. It does that under certain choices for the recurrent weight matrix. Easiest one is when it's just the identity, so that each neuron has recurrence onto itself, but onto nobody else. If you perturb that a little bit so that nearby neurons have recurrence onto one another, it behaves indistinguishably. But if you add lots of stuff off the diagonal, then you start pushing your way up here. Okay. Now, what's important to know about this graph is that for a linear recurrent neural net, as soon as this variable delta gets bigger than 0, it goes unstable. Okay. So we can go all the way up to here before it goes unstable because of the built-in normalization. Right. Now, if I choose this recurrent weight matrix in the right way, then I can go way up here and it's still stable. Right. But if I start adding random values off the diagonal, I can analyze this. And what I find is that there's a signature of the breakdown of stability, called critical slowing down in dynamic systems, basically is the spiral attractor regime. And that happens at about the same place that the fixed point starts to deviate from that normalization equation. And we can actually derive a closed form expression, the dash curve, for approximating. So the blue symbols, onset of critical slowing down, the red symbols, loss of normalization, the dash curve, analytical approximation for all of that. This looks like a big difference, but it's actually on a log scale, so it's pretty close. Okay. And so that's actually very powerful. Why does it matter? Well, I already alluded to some of this. For AI and machine learning, it helps us avoid exploding and vanishing gradients. Having this normal is great. Vanishing gradients occur in part or in large part because of this loss of the relative activities of the different units on the networks because of the signals, right? So we don't get stuck in that gradient regime, but because of the activities, relative activities maintained, as long as we're in that stable regime, right? And as I mentioned earlier, you can't train a neural net that's unstable. So in machine learning, the workaround is to introduce some hacks. And this is common, for example, with artificial recurrent neural nets like LSTMs and GRUs, that what you do is you put a threshold on the gradients. And if the gradients exceed that threshold, you clip them. Okay. And that helps to prevent instability from messing up the learning. But it's an arbitrary hack, arbitrary hyperparameter. If that threshold is too big, then it will still be unstable. If that threshold is too small, then, well, you'll avoid instability, but it will train really slowly. Right? What I'm going to show you is that with the built-in recurrent normalization in organics, we can achieve comparable or better performance without any of those acts. So no gradient. In neuroscience, failures of normalization have already been shown to be associated with epilepsy, autism, amblyopia, depression, and schizophrenia. Critical slowing down has been reported as a reliable marker for seizure onset, but hasn't been, you just measure how long it takes to get to this, to a steady state, right? But hasn't been measured for these other disorders, but the theory predicts that those things should go. All right. The theory also predicts when you hit that critical slowing down regime heading toward instability, that there should be increased sensitivity to noise. Right? And this has been observed by my lab in collaboration with Marlene Berman in autism. Right? That the trial to trial variability is within an individual. Okay, so here we are, as promised, a little bit about machine learning. Think of this as like a convolutional neural net, feature maps, weighted sums, some rectification and sigmoid output nonlinearity, right? Weighted sum, output nonlinearity, feed forward to the next level. Right? What have we done? Well, so far, we replaced that with this weighted sum, an output nonlinearity that is a recurrent normalization. Up to the next level. Okay. All right. So when we do that, and run it on some benchmarks, MNIST, go to the spine, results are at the bottom there. That's boring. So this is sequential MNIST. For those of you who don't know what this benchmark is, the MNIST data set is a bunch of handwritten digits, 0 through 9. It's a benchmark that Jan LeCun built convolutional neural nets for recognizing handwritten zip codes for the stuff. Sequential MNIST is a silly benchmark. You take one pixel at a time. You go scan line order, okay, and then try to classify the 0 through 9 digits. All right. The reason that it's silly, like you couldn't do this, I couldn't do this if you gave me one pixel at a time. But the reason that it's interesting is it forces the machine learning system to have some working memory. You have to hold on to information for enough time in order to combine it with other information later to distinguish, let's say the curve at the top of a 2 from a 0, right? So LSTMs, sequential MNIST and computed sequential MNIST performance there, we get comparable performance. But importantly, to get this to work at all with an LSTM, you need to throw this arbitrary gradient back at it. Knocking up our sleeves. And by the way, this isn't optimized at all. This was our first attempt at it. It just works. No tweaking, nothing. Right? This is the speech equivalent of MNIST. It's a data set of audio inputs in which a bunch of different speakers are saying 0, 1, 2, 3, 4, 5, 6, 7, 8, 9. All right. And one of the standard ways to apply machine learning to this is you first compute a spectrogram and then provide that as input to the learning system. And we're comparing ourselves here with other recurrent neural nets. And again, we're, at or beating state of the art. State space models for the aficionados can do better than this. And in fact, the Mamba state space model applied to the raw 8 audio does even better. Okay. And we're busy at work on that right now. Okay. There's a version of organics. I told you to do the analysis, you linearize around the fixed point, right? When you do that, you end up with something that's in the form of a state space model. So there's the linearized organics are state space models with built-in recurrence. Now, what haven't I done? Well, on like the first slide, I said is for every feed forward connection in the brain, there's a feedback connection in the brain. I didn't include that yet, so let's add it now. All right. So now I have, say, V1 here, V2 there. I've added another cell type just for convenience. Don't worry about it. But within V1, I'm doing the same computation. Input drive, weighted sum of the inputs, input gain, recurrent drive, weighted sum of the outputs, with a recurrent gain that depends on an outer loop, right, that shuts down the recurrent amplification to do the normal symptom. Feed forward to V2, same thing, except that now I'm also adding a feedback drive, which is a weighted sum of the outputs of these guys get added to the membrane potential. Okay. And while I do that, I have to take a copy of this and have it contribute to the normalization pool to keep everything stable, and so that the output responses here and here both follow the normalization push. What's that? All these different symbols? Yeah, so if I wanted to actually write out the full dynamical system, it would be an equation for each of these. These are different cell types in the brain. There are actually hundreds of different cell types, okay? So the proposal is that each of these is a different cell type that's performing a particular form of computation. Each neuron in this circuit performs a slightly different computation, okay? Which makes sense. There wouldn't be hundreds of cell types if they all do the same thing. And I will, time permitting, hypothesize a relationship between each of these cell types and identified cell classes in the brain. Okay. All right. This copy of the feedback has to go here and contribute to the normalization pool. That's for those of you who know this. That's consistent with evidence that surround suppression in V1 partially comes from feedback. So there's evidence that that exists. All right. So then we add noise to this because neurons are noisy, right? The noise in the brain mostly comes from stochasticity of synaptic release, right? So an action potential releases an axon terminal. There's about a 10% chance of neurotransmitter being released. That's where most of the noise comes. Beautiful work that was done by Chuck Stevens. Right. So we add noise, and then again, not me. For those of you who know me, I didn't do this math. What I do is I said to my collaborator Stefano and the graduate student we were advising Siobhan about 5-6 years ago. I said, "So can you guys derive the power spectrum?" I said, "Sure." 2 years later, he came up with the answer. Along the way, Siobhan, actually, this is true. He reproved a theorem that had first been proven by Isaac Newton, but he didn't know that it had been proven by Isaac Newton. So there's some serious math here. They published a math paper on this that has like 100 equations in it, some of which are a full page long. The bottom line is that you can do it. Okay. Conceptually, it's pretty straightforward. You say, okay, I know what the fixed point is. Because I know what the fixed point is, I can linearize around the fixed point, and then I can derive the power spectrum and cross spectrum. All right. So everything on the left here is analytical. It's not simulation. You just evaluate it at closed form expression, right? That's a big deal, right? Because if I had to compute these results through simulation, right, what I'd have to do is I'd have to run the recurrent neural net with one sample of noise, compute the power spectrum, cross spectrum, run it again with another sample of noise, repeat that about 1,000 times, average them all together. And then if I wanted to fit data with that, then I'd take the mean squared error with the data, take a gradient step to adjust the parameters, and do the whole thing over again. We'd be dead before we'd gotten it. Right? But now, because it's a closed form analytical expression, we can compare it directly against the data. Contrast response functions in V1, V2, these experiments from different labs, published different papers. Power spectrum in V1, the coherence between V1 and V2, local field potential compared with experimental measurements of these communication subspaces in the subspace idea is you take a subpopulation of neurons in V1 and a subpopulation of neurons in V2, and you say how well can I predict the firing rates of the V2 neurons as a weighted sum linear combination of the V1? Okay, and how many dimensions do I need? And this is the number of dimensions here. So in spite of the fact that there are dozens of neurons being recorded at once here, the dimensionality is low. That's what you see there. And you can do that for subpopulations of V1 to V2 neurons, or one subpopulation of V1 neurons to another subpopulation of V1 neurons. And you see these differences in the ability to predict how well can you predict the fire rates of one subpopulation from the other as a function of the number of dimensions. Right. Coherence is like correlation broken down by frequency. Okay, so we haven't tried to fit any data yet here. Again, this is sort of just right out of the box. It's pretty good, right? There are a handful of parameters that matter. If I were to change all the time constants on those differential equations, then it would shift all of these frequencies to low frequencies, for example. That's it. All right. So the theory also predicts things that haven't been measured, right? So this is the coherence from the previous slide. This is that prediction performance of the communication subspace that's broken down by frequency. This measurement hasn't been done. The theory predicts that these two things should be aligned with one another. And this is the dimensionality of the communications that we predict should be inversely related. All of that comes about because of the normalization. If I set all the normalization weights to 0, so that in the denominator of the normalization equation, that W times Z goes away, because W is all zeros, then I'm just dividing by the constant sigma squared, no normalization. All of this stuff disappears. There's no gamma peaks in the power spectrum. They don't shift with contrast in the way that you see experimentally. There's no gamma in the coherence spectrum, nor in the communication. It all just goes away, right? The intuition for it is, or the way that I sort of roughly think about it, is that, with the normalization in there, right, what's happening is there's this recurrent amplification, and then the outer loop shuts it down. Okay? So you can imagine what can happen. This is happening fast. This is happening a little bit more slowly. It's at certain frequencies, you're going to get sort of a resonance, right? That this inner loop is going to happen fast enough, not so fast that it can't keep up, but fast enough that the outer loop is slightly lagging behind, and you get these peaks. Where does attention come in? There's a little demo from back when I was doing imaging work of ending right, left, and seeing activity in the brain go left, right, left, right. There are two different ways in this theory that we can modulate attention. That is, modulate the sensory activity based on attention signals. Going back into sensory cortical areas. This one is something that John Reynolds and I published about 15, 16 years ago. If you change the input gain, right? Remember, input gain times input drive. Change the input gain, then that causes shifts in the contrast response. Make that input gain bigger and bigger, and you shift to the left, or for any given contrast, the activity gets stronger, right? But we can also now play around with the feedback gain. Right? So we have this feedback gain times feedback drive, right? And you get something that's pretty similar, but you can see that it actually, the shape of the curve also changed. Here it's just a pure shift. Here it's a shift into kind of amplification. A hypothesis is that this corresponds to what psychologists for decades have been calling exogenous attention feedback, right? So what I mean by that? Exogenous attention, look at my face, attend to the right, attend to the left, okay? Right? It's a symbolic cue or an instruction. Hold a representation, presumably in prefrontal cortex, that percolates back and modulates the activity. What's exogenous attention, if there's a bright flash or something at a particular location, the filter automatically grabs your attention there involuntarily and There's no control of it. You can't shut it down. I can tell you to try to ignore it and it would happen anyway. Okay. Functional connectivity. Right. So if I increase the feedback gain, so now I'm just going to build a model which is V1 and sending projections to two different brain areas. I'll call them V4 and V5. V5 is MT, motion pathway, form pathway. Okay. I'm actually not doing anything interesting with them though. It's just, the output of V1 goes to a weighted sum here, goes to a weighted sum there. The weights are the same. Okay. And then there's feedback. Feedback gain times feedback. Same. All I'm going to do is increase the feedback gain on one of them or the other one. Right. And what ends up happening is that the communication subspace shifts. So if I boost the feedback gain from MT to V1, then there's strong communication from V1 to MT. V5 is a synonym. If you're in London, you call it V5. In the United States, you call it MT. But versus if I increase the feedback gain from V4 to V1, I increase the communication. So there are now lots of observations, particularly from neuroimaging, but also from multi-unit recording simultaneously in different brain areas, that in spite of the fact that the actual hardware, the axons aren't growing quickly, changing quickly, but by changing task demands, right? There are lots of observations of dynamic changes in the functional connectivity, how strong the functional connections are, even though the anatomical connections don't change. And to me, that has always been a mystery. How could that happen? Right? The idea here is we have at least one explanation of that just by changing the feedback from the higher area to the lower area, boosts the communication book. Think of this as all recurrent loops. Briefly, working memory, we've got that too. I already kind of explained the idea of long-term dependencies. You have to hold some information online until other information becomes available, combine it in order to get an answer, like distinguish one input from another, or what you're doing right now, listening to me talk. The sentence gets resolved later in time. You can do that easily in linear recurrent neural nets or gated linear recurrent neural nets. The key thing is that it's very hard to keep. So that's what's shown here. This is a linear recurrent neural net in which the recurrent weights are ever so slightly larger than one. But the spectral radius or the largest eigenvalue of the recurrent weight matrix is ever so slightly larger than one, and in no time it expands. Okay? This is the organic circuit with the same recurrent weight matrix. And you can see what happens is that, the recurrent weight matrix, the recurrence tries to boost it, but the normalization kicks in and shuts it down. So you get these little low frequency oscillations, but importantly, the ratio of the responses. That's another way to look at the stability issue and the importance of normal. Sequential activity, we got that too very briefly. I promised to show you, so I'll flash it up. These different symbols that I would write out in the whole system of equations for the dynamic system, or we can hypothesize that they identify with particular identified cell types in the brain. We can hypothesize a certain layout of the computation on the dendritic tree. Pyramidal cells in the brain have this crazy shape. When I first learned about this, I was always scratching my head. Why would you have a cell with a shape like that? Dendritic tree down here, all the basic. Turns out the feedback signals mostly come in on these distal branches of the apical dendritic trees. I'm going to put the feedback drive there. The bottom-up inputs come in on the basal dendrites. I'm going to put the input drive there, and then I'm going to put these gain controls closer to the cell body. So we get input gain times input drive here, and then up here I'm going to get feedback drive times feedback gain, and the recurrent drive multiply the recurrent gain, and that's the formula that we need. And how do you actually do that with neurons? Some of you already know this. You can think of a neuron as an RC circuit. The capacitor is the lipid bilayer, right? The synapses are variable conductance resistors, right? There's a sodium-potassium pump that sets up an electrochemical gradient between the inside and the outside of the neuron, okay? If I were to, which pumps sodium out, potassium in. If I open a little hole in the membrane that's selective only for sodium, sodium ions will rush in and deep. If I open a little hole in the membrane that's only allows potassium through, potassium will rush out and hyperpolarize. That's exactly how synapses work. Neurotransmitters released at the synapse. It binds to receptors on the postsynaptic cell. That changes the conformation of proteins across the membrane that are selective for particular types of ions, sodium or potassium. All right. So we can think of this process. You know, there's a electrochemical gradient that we model as a battery, and a variable conductance resistor that corresponds to whether these channels are open or closed. If I want to do addition and subtraction, what I do is I change the excitatory, the sodium and potassium channels in a push-pull way, so that if I increase the conductance of this one, I decrease the conductance of that one, and that way I get a current injection with no net change in conductance. If I want to multiply and divide, I do the opposite. I do push-push. I change both of them in a ratio, so increase both together in a particular ratio, depending on what the results are. Then I'll get a change in conductance with no net current. Conductance will just flatten out, right? The voltage versus current, right? V equals IR, right? If I change the resistance, I'm changing the slope of that, right? Okay. And you can elaborate that into this form to write out this one. And there we are. Okay, so I finished in almost exactly an hour.
What's Maxwell laws?
Well, I mean, I didn't write the equations for you. They're in the papers. But it's basically what I think the principal cells are: tau divided equals minus y plus input gain times input drive plus recurrent gain times recurrent drive plus feedback gain times feedback drive. And then the key thing is that one of those recurrent gain terms has to be of a particular form so that normalization is the fixed point, right? And that computation, you can also imagine a biophysical implementation of it, but I left that out. And through those other solvents. All right. I could flash up the equations for you if you want. Okay, we can do that afterwards. We'll let everybody else, stop. Thank you very much.